you for allowing me the opportunity to present. And today we're going to talk about some of the miniaturization of protein-protein interaction, high-throughput screening in 1536 well plates that we've done here at small molecule screening facility at UW. So first up, I'd like to introduce our facility and some of the folks that work here. So Professor Michael Hoffman is our faculty director. Myself, Song, and Ken work on the high-throughput screening aspect of the lab, and then Scott, Spencer, and Ken Sadashir work on medicinal chemistry and computational chemistry here at the facility. And we've been around for quite a bit. So we were established in 2003. We're a fee-for-service lab, and if you want to learn more about our services, this is our website. So the first question that we usually get when we do these kind of presentations on campus or off campus is essentially, why small molecule screening facility? What are small molecules? So a small molecule is defined as a low molecular weight organic compound that may perturb a biological process. So usually these compounds are under 900 daltons. And why they're important is that most drugs that we have in use today are small molecules. So you can think about the small molecule as an exogenous ligand that binds a target protein and somehow affects its function. I do usually like to give a little bit of the background on the methodology of high-throughput screening, how the field came about, and some of its history. So the origin of high-throughput screening is really in the mid-1980s, and the origins are in natural product screening for antibiotics. Originally, labs were doing disk diffusion assays or liquid broth assays to inhibit growth of specific microorganisms, and the throughput was quite low. So you're talking about one extract is usually one test. On the order of milligrams of the compounds were used for these experiments, and even the best labs were only doing 20 to 50 compounds per week. So by mid to late 1990s is when you start seeing a lot of automation take place on this. At this point, compounds got standardized as being stored in DMSO, multi-well plates came on the scenes, and labs were doing about 10,000 compounds per lab per week. And the primary interest at that point was oncology targets. And then around 2000 to today, there's been a resurgence of going back to natural products and screening for antibiotics. But really, it can be defined by further miniaturization. So we're going to 384 well plates, 1536 plates, and now even lower formats. So at the heart of our facility, besides all the laboratory automation instrument, is our small molecule collection, which is can be called a compound collection. So we have about 500,000 compounds, all of which are stored in either 384 or 1536 well plates. So here you can see an actual compound plate with the different compounds in it. The majority of our compounds are what's known as diversity compounds, with the largest single collection that we have is the NIH's MLPCN library, which is 330,000 compounds. In addition to that, we have a number of smaller libraries, so all known FDA drugs, kinase inhibitors, anti-infectives, some natural products, etc. So really, for us as an academic facility, the question goes is, we have this wonderful collection of compounds. How do we screen them for disease-relevant targets and do drug discovery in an academic setting with academic budgets? So I'm going to talk to you about a couple of examples of protein-protein high-throughput screens that we've done for the laboratory of James L. Keck, who's faculty in biomolecular chemistry here at University of Wisconsin-Madison. And first, we're going to walk through a paper that came out in 2016 with the work that was done at the facility. And this is kind of one approach to doing high-throughput screening. So this was a fluorescence polarization screen for the protein-protein interaction, which is a Fanconi anemia. So this was done. Well, I'll show you the screen setup a little bit later. So this is the actual setup that we had for the screen. We screened about 75,000 compounds in 384 well format using tip-based liquid handling. So in total, we used about 2.4 liters of the protein solution for the FT assays. And this screen was fairly expensive because of the amount of tips that we used, and also fairly large dead volumes. So 5 to 10 mil per day of the protein solution was going to waste just on dead volumes. And here you can see this is the setup for 25,000 compounds. So these are all the tips that were being used, and Actually, the tips don't go into a landfill. They end up getting burned and incinerated because they're biohazard waste and the FX. So after the experience of this HTS and the data that we got, there was really a lot of interest from the Keck group to screen more compounds, but to do it with a more manageable budget. So this is a paper that came out of their lab in 2017 on a high-throughput screening strategy to identify inhibitors of SSB protein-protein interactions in an academic screening facility. And so the goal that we set out to do was to minimize cost in protein use and to come up with a cost-effective means of screening 100,000+ compounds. So just to give you a little bit of background, is one of the secondary screens that we used for the initial FP screen was using alpha screen technology from Perkel and Elmer. So this is a bead-based ELISA replacement, where essentially you're doing a sandwich-based assay to capture an analyte using two beads, which when one of the beads is excited, it emits oxygen and produces visible light. So this is a very rapid assay. It's not labor intensive at all, can be done in about an hour, and it's scalable to low volumes in 1536 well plates. So why 1536 well plates? Obviously, the answer is, because of size, but really on our end, it's because of volume. So here we have a 2-scale drawing of the various well plate formats, so 48, 96, 384, and 1536. And volume scales accordingly. So by using 1536 well plates, we can conserve protein, reagents, miniaturize, and the cost savings are actually very significant. They're an order of magnitude, especially on the detection reagents such as alpha-lysa beads or luciferase, anything like that. So for a 384 well plate, we're using anywhere from 20 to 60. For 1536 well plates, we're using somewhere between 3 and 5. Now, for anybody that's worked with 1536 well plates, doing this with tips is actually extremely expensive and cost prohibitive because you generally need proprietary tips. So at that point, our lab was fortunate enough to, through various sources of financing departments and grants and the facility itself, acquire an Echo 550 that we bought, and then we also have a Mantis instrument and a BMG Ferrostar plate reader. So the setup for the screen was to use nickel chelated and streptavidine donor beads for the alpha-lysa assay, and to miniaturize it using a synergy of these three instruments. So we used an Echo 550 for compound additions into 1536 well plates. So we're adding 10 nanoliters. For the master mix, which is the protein and alpha-lysa beads, we were using 3 microliters per well. And then the detection was done on a BMG Ferrostar. And an important note here is because of kind of the Mantis's ability to use very low dead volumes, this helped us conserve reagents incredibly. And also we completely eliminated tips, obviously, using this methodology. So what is the kind of end result of it is that the protein use was scaled down by about an order of magnitude. So to screen a roughly equal size library, compared to FP, we only use 240 ml of the protein solution. And for the bead use, so while the manufacturer recommends for 25 microliter reactions, we're getting about 10,000 data points per 5 mg of beads. With the way that we were able to scale it down in 1536, we're doing close to 300,000 assay points for the same reagent. And most importantly, if you've ever made protein, that's a very labor-intensive process, is that the complete use of protein for this screen was only 5 mg compared to what was estimated to be 1 gram for the FP screen. And since this paper came out, we actually now screened in excess of 500,000 data points for this target, and we're screening multiple other targets with this and other labs. As far as data quality and kind of what we came up with, so we screened 73,000 compounds with 9 hits coming out that are potent and selective. Z prime scores were all above 0.5, so the data quality is actually very, very good in this methodology. And so to kind of further continue on this topic, using a combination of the Echo and the Mantis instruments, we've done alpha screen for several other targets. We've also done a number of cell-based reporter gene assays using nanoglow as a luciferase detection reagent, and some biochemical screen for metabolic enzymes. And actually here is a picture of a recent paper, lab of Xinyu Zhao, also here at UW-Madison, reporting some 1536 well screens that we've done using iPS cell-derived neuron progenitor cells, actually. And so with this, I'd like to give you my contact info in case you have questions about assays or targets or anything like that, and just give you kind of a broader overview that, so small molecule is just one part of this larger drug development core here at UW. And all three of our facilities, so the small molecule screening facility, the cancer pharmacology lab, and the medicinal chemistry lab, offer our services to more or less anybody in the university system and outside of it for drug development needs. Thank you.